Hello everyone. At the end of class today, we were trying to wrap up an activity talking about how we would produce cortisol. We rushed through the explanation, and so I thought it'd be a good idea if I went ahead and drew out the pathway here. That way you would have it, and you would have the version of it that I would want you to know for the exam. Remember what happened in this scenario. You were all, as you remember, horribly scared. You probably blocked it out, so you probably don't remember it. So you were scared because of that image, and then as a person, right, you received that through your eyes, and that sent a signal to your brain. So let's expand this area here, and we'll draw a very poor drawing of a brain. And in the middle of this brain, there is that gland, remember, called the pituitary gland. It's one of the master regulators of the body. It produces lots of hormones. We didn't go into the mechanisms of how the pituitary gland is stimulated, but it's stimulated by another hormone. But the point I want you to take out of this is that the hormone that is now released from the pituitary is this hormone called ACTH, which stands for adrenal corticotropic hormone. But on the exam, if you know it as ACTH, that would be sufficient. As it's released from the pituitary gland, it enters the circulatory system. And at some point, it's going to reach the kidney. But it's not the kidney we're interested in. It's the adrenal gland that sits on top of the kidney. So let's expand the adrenal gland, at least one of the cells. The adrenal gland will have a receptor that ACTH binds to. In fact, let's draw the cell a little larger. As ACTH binds to this receptor, it turns out to be one of these receptors that's linked to a G protein, and it's going to activate this G protein. This active form of the G protein will have GTP bound to it. In the podcast, the original podcast, I talked about the alpha and the gamma subunits of the G protein. This is the alpha, and that's really the main one we're going to talk about for this class. So we can think of this as the alpha part of the G protein. But the part I want to talk about right now is that it activates, the active G protein will now activate adenocyclase, which is also from our podcast, original podcast. As this is activated, it will produce cyclic AMP from ATP. Cyclic AMP will activate protein kinase A. Protein kinase A will, through various mechanisms that we didn't talk about and we're not going to, will synthesize cortisol from the mitochondria, which we'll just show as this blue circle. Cortisol will now be removed from the cell through mechanisms that we don't need to talk about, but it will enter into the circulatory system. Now, this cortisol is going to move through the circulatory system until it reaches one of many target cells. But one of these target cells could be found in the heart. So let's take a cell from the heart. Cortisol will then move freely through the membrane. It will bind a transcription factor, which I'm just going to abbreviate TXN FAC. So let's write that down here. TXN FAC is equal to a transcription factor. This transcription factor will move into the nucleus, and I'm going to make the cell bigger now so we can have a nucleus. So the transcription factor with the cortisol bound to it enters the nucleus, and in the nucleus it will turn on genes necessary to increase the heart rate or break down glycogen if we're in the liver or some other tissues. But right now we're talking about how it's affecting the heart. But cortisol will have other targets that it will find and alter which genes are turned on or off in the nucleus so that it can respond to this initial stress event. At some point, the stress leaves and you're no longer stressed by that horrible image you saw. And now it will go back to the brain, the cortisol, and at high enough concentrations, this cortisol will inhibit the production of ACTH and therefore inhibit any further production of cortisol. Okay, the only other thing I want you to know on this picture here is that ACTH, since it doesn't go through the membrane and it activates one of these G proteins, you would assume that it is a large hydrophilic or you might say lipid insoluble protein, because it is unable to easily move through the membrane, so it signals its receptors on the outside. This example also provides us with a signal that is small and hydrophobic. So cortisol here is going to be small and hydrophobic, or you might say lipid soluble, because it can first enter into the cell and find its target receptor inside the cell and enter into the nucleus. Small hydrophobic or lipid soluble signals can move through the membrane to find their targets. Large hydrophilic, or you might call them lipid insoluble signals, can't do that. They have to have a signal on the outside of the cell. Okay, so this is what I would want you to know about the wrap-up from the activity we finished in class today. In our next class, we're going to talk a little bit more about cortisol and think about this pathway. So become familiar with this pathway so we can apply it to the next question we have involving cortisol in our body. All right, let me know if you have any questions. If not, I'll see you in class. Bye.